Hey, David. Good to see you. I'm going to wait a second, see if anybody else joins us. Okay. Happy Thursday. And we were talking a little bit about our assignment, if you had any questions on it. One of the things that I didn't define the other day is something called, if I can spell, heterotroph. Okay, and I just posted it up there, Heterotrophh. That PH makes an F sound, heterotroph. Okay, that's something that's on your assignment. It's basically any living organism that can't produce food for itself. So heterotrophs, it would include herbivores, omnivores, carnivores, animals that we know, that we think of, because that includes humans. So we eat other organisms to get our protein and our energy. We cannot produce our own food or energy by ourselves. Majority of things that we know of are heterotrophs. If you named an animal, a human, a horse, a dog, a cat, a bunny rabbit, a bird, they're all heterotrophs of some sort. Nick, good to see you. Glad you joined us. Trying to think what else might be on that sheet that we didn't talk about. We talk about guard cells. Guard cells are found on the underside of a leaf. They basically control how big the opening is on the other side of the leaf that lets oxygen in, carbon dioxide out. Basically, they're like a gatekeeper. Think of a valve. When you turn the faucet on, you open the valve up. The wider you turn it, the bigger the opening gets, so the more water can come through. Guard cells are just like that, except they're on a leaf. And on that leaf, they open to bring in carbon dioxide, and then they also open to release oxygen. Okay? And so there's a couple things we didn't talk about the other day on that sheet, if you didn't figure that out. You should remember what organelles are. Those are the little structures inside a cell that we talked about at the beginning of the year. One example of that is the mitochondria. Mitochondria is where most of the cell's energy is produced. We talked about lactic acid fermentation. I wish Mr. Fosnight was able to talk about that, especially when you're doing athletics and sports. It's basically a different way of getting energy. And so let's say that you're doing a sport, if you're wrestling, if you're running long distance, after a while you start, whenever you're doing something physical, and Nick, you probably understand this from playing basketball. After a while you feel that burn, get that burning sensation in your muscles. That's actually a secondary way that your muscles are trying to get energy. It's called lactic acid fermentation. It's a cellular process. And so when your muscles can't get enough oxygen quick enough, which they usually can't if you're really exercising at a high level, like basketball, where you're not jogging, you're running a lot. And so you know exactly what I'm talking about. And so that muscle in your leg or your arm isn't getting enough of that oxygen it needs, and so for energy. And so it is using something called lactic acid fermentation, and that's where that burn comes from. Not necessarily fun, but your body's finding a substitute for that. That pretty much should cover everything on that worksheet. We know glucose is just sugar, and that pretty much should cover everything. There's only 8 questions. Don't be afraid to go live, or don't be afraid to message me either here when we're live or on Google Classroom. You can message me, or you could even message me by email. You can send me an email. So you have multiple ways of getting a hold of me if you have a question about something. All right, so we're going to change gears a little bit. We're going to we're going to talk about the scientific method, okay? And usually this time of year, we have multiple things going on at once in school. We'd be working on our gardens, preparing the garden, starting plants in trays, talking about plant stuff, and we'd also be starting to go through our another cycle of doing experiments, and these are experiments that you choose to do. So we are going to start into that again. Eventually what this is going to evolve into is you doing experiments at home. Okay, so if you haven't said hello, please do. 1, 2, 3, 4. Okay, I think we got everybody. If you haven't said hello, please do so we know we're here. But let's talk about the scientific method and how does it start off. If you know what the first step in the scientific method is, go ahead and type it into the box. But I'm going to tell you how, I'll give you an example of how we use this in the real world. Right now, I am struggling with what I believe to be poison ivy. I got it on my arms, got it on my lower legs, and it keeps showing up in different spots. And it's been weeks, and I'm frustrated, and I'm really itchy. And so in a real sense, something like that happens in the real world. We go, okay, we can turn that into a scientific method problem where it's a modified, we change it a little bit, right? But we actually do use part of the problems that we use in the scientific method. So the first step of the scientific method is simply this. It's not typing. Okay. And that is to do this. Ask a question. Okay. So my question is, what is making me itch? Is it in fact poison ivy? Is it poison oak? Is it poison sumac? Is it something else that I just don't understand? That's my question right now. I'm just giving you an example. So once we have a question, we want to learn as much about the subject as we can. That brings us to our second step. Our second step is doing background research, okay? I don't know if you guys remember this from earlier in the year. Okay. So what are some of the ways that we would be doing background research? For me, with my poison ivy, I've been getting on my phone, getting on Google, and saying, what can cause itchy skin, right? Okay. And so you find things like poison ivy, could be that, poison oak, could be that, poison sumac, could be that, but there's other things that cause itchy skin too. An illness called shingles, psoriasis, there's in bacterial infections you can get, something called scabies. I was looking at it all thinking, okay, this just keeps itching. What do I have? Why is it acting differently? And so part of my question is, because I've had poison ivy a lot in my life, I'm asking, why is this round of whatever I have acting differently than when I've had it in the past? Because I grew up playing in the woods behind the house in my neighborhood, and I spent most of my life with poison ivy growing up. Pretty much had it just about every year, and learned to live with some amount of itching, learned how to clean it, and how to not itch it, how to not scratch it, and keep it from spreading. And so I have some of my own background knowledge, because your background knowledge can be in your head. It can also be in somebody else's head. I could ask Mr. Fosnight a question about it. Or let's say I had a question about something that he's an expert on, about lab procedures. He spent years working in a lab. He's an expert on lab procedures. If I had a question about a lab procedure, I'm going to go to somebody like Mr. Fosnight. So we can use the knowledge that's in our head. We can use the knowledge that's in somebody else's head, or we could use our phone and go to Google or another online resource and use that as a possibility of how to find out that knowledge. And so we've asked a question or multiple questions, but we want to stick to one in this case, but we can do multiple experiments. We've done our background research. What do I know? What do my friends know? What can I find online? Okay. The third step of all that is doing what? Is creating a hypothesis. Okay. Excuse me. And now if I was in public, I'd be coughing into my elbow. So a hypothesis is nothing more than what, guys? Somebody tell me what a hypothesis really is. We talk about this all the time. You talked about it in elementary school. What is a hypothesis? The first word starts with E. The second word starts with G. An EG. That might spur somebody's memory. I'll give you the first word. Educated. Educated what? So that hypothesis is an educated guess, okay? We're trying to predict what will happen if we do an experiment. Why do we do an experiment? Because we're trying to see if, we're trying to answer that question if we're correct. We're trying to see if we're correct about the question that we asked. Is this poison ivy? Is this poison oak? Is this a different type of infection? And so for me, what I'm dealing with, I'm asking a lot of questions and trying to process each one. And if I don't get the answer that I'm looking for, if I fail, which in experiments we fail lots of times, we go back and we ask another question, or we change our hypothesis. And I'll get into that a little bit more in a second. So a hypothesis is a prediction of what will happen in our experiment, okay? And we have to identify, we have to have controls. That means certain things need to be the same all the time in each experiment. And then we have to have one thing that changes, which is called a variable. A variable, we use that variable to see, okay, I tried this experiment this way and I didn't get the result that I wanted. Let's change that one variable. It could be how much water that you're using. Maybe you need to use more water or a different solution. Maybe water doesn't work, so you're going to use vinegar. Whatever your experiment happens to be, you change a variable. It could be quantity, it could be what you're actually changing. But the important thing is you only want to change one variable at a time. Because if you do end up getting the result that you want and you've changed 2 or 3 things at once, do you know what caused that to change? And it's impossible to know what caused you to get the result that you wanted to do if you're changing multiple variables. Okay, so one variable at a time, you check it. If you fail, you go back, change that variable. So the hypothesis is that educated guess. The next step is to actually do the experiment. Okay, this is where you're actually going to be doing the experiment. You start off by, you need a supply list, right? What supplies do I need to complete this experiment? Do I need a beaker or some sort of a measuring cup? If you're doing it at home, you can use measuring cups, you can use teacups, anything that you know, if you know the volume of it. Now, you don't have stuff like this running around at home, but Mr. Suriani, you have it at home. I went to school and got a bunch of supplies. Unfortunately, sometimes this ends up being a place to keep my pens. But anyways, we need to know what supplies we need and our steps. Remember, we're writing steps. What do I need to do first? Okay, what do I need to do to make this experiment work? This is really good practice that make you guys into really good scientists. Okay, when you have to write your own instructions, and we do something interesting. We did this in the beginning of the year, and we're going to do it again, where we're writing our own experiments. And think of it like this. If we did it with food, right? And I want you to think about that too. If you were going to make toast, or if you were going to make chocolate chip cookies, or if you were going to bake a cake, there is a set of instructions to do that on the box if you use a box. What if you're not using a box? Do you go online and get a recipe? Do you just goof around with it and try to get it to work? Do you listen to what your mom or your aunt or your grandmother or grandfather or dad or whoever, men cook too, what they told you to do. Either way, you're getting a set of procedures, okay? In this case, we're going to be writing them down, okay? The whole point of this is you guys are going to be doing an experiment at home using household stuff that you have around the house, everyday stuff, and we're going to test to find results on various things, okay? So step number 4 is doing the experiment. Let's go through those again. We ask a question. That's step number 1. We do background research. That's step number 2. We come up with a hypothesis. That's step number 3. We do the experiment. That's step number 4. Step number 5 is we get our results. Now, Mr. Fosnight and I have talked about this a lot. How many times we've seen people come up with a really, really cool experiment, and we're thinking, oh, that's going to be, I don't know what the answer to that is. You know, Mr. Fosnight, do you know what I don't know what the answer to that question is either. All right, well, we're excited about it. We want to know what the results are going to be. And usually the students are excited too. And so they do the experiment, they go through it, and they have a blank piece of paper when they're done. They didn't record any data. And they didn't take that data that they recorded and make it into a graph. One of the things I sent home with the 6th graders was analyzing data worksheets. Okay, if you went and picked them up, you know what I'm talking about. There's pie charts and bar graphs and stuff, and so you're thinking, oh my gosh, this is, you know, why is this stuff here? Because when you go to report your results, it's very easy to report your results with a graph or a pie chart or some sort of way to actually use a chart to release your data. And so that's huge, understanding how to do a graph. That's going to be a struggle, and we'll work through that when we get to that step. And the very last part is your conclusion, okay? So, and that means, what did you learn about your hypothesis? Were you correct? Were you incorrect? Did you learn something differently? Usually a conclusion says, our prediction was right, but the results were still not exactly what we expected, but we were almost there. Maybe that's what it was. We expected that 75% of the time we would get result A, but we only got it 60% of the time. And that's where you kind of say, what did I learn? What did I learn about this? Why was my prediction wrong? Why was my prediction right? And this is where you start saying, what did I learn that was different than what I expected to learn? And how could I take that information and change it a little bit and come up with something a little bit different? So your conclusion and interpreting that data and coming up with your conclusion will help guide you to either fix your experiment or help you ask another question. A lot of times when we get a conclusion from an experiment, it helps us to ask another question. We think, oh, this is the result I got. That's cool. Why did this happen? Guess what? That becomes a second experiment. Why did this happen? Let me test that. And so a lot of times experiments will spawn questions that allow you to do second and third and fourth experiments. Totally fails. What do I need to change to make it work? That would be in your conclusion. All right, so anybody have any questions? If not, I know I've gone on a long time, and I hope you remember some of that stuff. I will be posting some stuff for you guys to work on. Make sure that you're submitting the work that is already out there. Even if it's past the deadline, go ahead and do it, get it done. All right, what movie is my shirt from? Flux capacitor. It's Throwback Thursday, movie day. Any answers? No answers. Is anybody awake? Did anybody turn on their phone and go get a sandwich? Maybe. Okay. Hello. Goodbye. Nick, I hope you have a good day too. I missed you guys. I got to see David last week. That was awesome. And I hope I get to see you guys. Maybe I'll run into you in the grocery store. Who knows? Out and about. But I do miss you guys. David, get some sleep. All right, you guys have a great day. Don't forget, if you have any questions, even on the weekends, I'll be checking my email. I check it every day, multiple times during the day, even in the evenings. There's a couple students that seem to be doing their work at like 7, 8 o'clock at night, and some at 7, 8 o'clock in the morning. And so I check it multiple times. I hope you guys have a great day, and I'll talk to you soon.